Good morning, students. Good morning, everyone. Within 1 minute, the section will be start. YouTube Live and Facebook Live is also going on. Please check it there also. Who are facing in the trouble in admit, you can join in live section also. YouTube Live and Facebook Live is also going on here. Sir will join within 1 minute. The section will be also start within.
Hi Maheshwari, can you hear me?
Yeah, it is audible, sir. You can.
Maheshwari,
Yeah,
yes, can you hear
good,
me?
sir. Yeah, I can.
Yeah, yeah. Just check my screen also because I'm at a different site today, so just want to see the internet connection. Then we can start. Okay, so I'll share my screen.
Okay.
Let me know when you're able to see that. Okay.
Yeah, okay. Please on your camera also, sir.
Oh yeah, yeah, sure, sure. Definitely. Are you able to see my screen?
Yeah,
Maheshwari, can you see my screen?
network is, yeah,
Okay,
I
and
can
voice is also very
see a
clear.
screen. Your network is up and downing. Please check it, sir.
Okay, 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 okay. All right. What I can do. Give me 1 or 2 minutes. I'll try to fix it. Okay.
Students, please inform your friends who have not joined. Let them join as early as possible. YouTube Live and Facebook Live is also going on. You can watch there also. Even facing an issue in air means you can join me in YouTube and Facebook also. Please check it.
Maheshwari, can you hear me?
Yeah, I can hear you, and your PPT also visible.
PPT.
Sir, your network is not good. It is already in red symbol. Please check it.
Visible.
Yeah, now it is good. You can. Again, you had lost the net. Students within start facing some network issue. Within 2 seconds, the section will be start. Please inform to your friends also. Please don't miss the section. Okay. Within 2 seconds, please. You patience for 2 seconds. The section will be start. Sir, you can on your camera and PPT, sir.
Yes, my shooting. I'm starting in another 1 minute. Just on another laptop, I have logged in. So just taking out my presentation. Just give me one more minute, okay? I think now the
Yeah, okay,
connectivity
sir.
is good. Okay.
Yeah, now it is good. Sir, please unmute your mic.
Yeah. Can you see my screen? My screen?
Yeah.
Fantastic. Is my voice clear to
No,
you?
no, screen is not visible. It
Screen
is
is not
blank.
visible.
It is blank. Once close the, yeah. Yeah, now it is visible. You can continue, sir.
Okay, fantastic. So, very good morning to all of you, and my sincere apologies for slight delay because of some connectivity issue. But we are all set now and ready to start. So, very good morning to all of you, and welcome to an online class on drug discovery development course, which we have started almost 10 days ago. So, today, what I thought was we will actually do. Kind of a recap, okay? Because we are going to have an assessment next week on Tuesday. So before that, we need to have a recap as well. So what I'm planning to do is tomorrow I'll cover the remaining topics, especially on what all topics are remaining in our course. We'll try to complete it tomorrow. And today, let me start the revision from where we started. Okay, especially what we have discussed within this week, I think would be completely fresh in your mind. So I may not have to do any revision for that. But when we started this course a couple of weeks ago, I think that we need to recap once. So I'll start from there, and then I think hopefully I'll try to touch base on most of the topics in first week what we have covered, and then tomorrow we'll talk about new topics, and then on Monday we will just revise the entire course. If you have any questions, we can clarify those questions, and then you'll be ready to go for the assessment. Okay. So the course which you all have been attending, as you all know, is a drug discovery and development, an overview of the entire drug discovery development field. Okay, it's and this course again at this point in time would like to congratulate Andhra Pradesh State Skill Development Corporation for their very great initiative of conducting this online training programs, which is really beneficial to students across the state of Andhra Pradesh and even to students outside of the state across country. So, would like to congratulate Andhra Pradesh for their such a novel initiative, for skill development of students. And so, what we in the first class we talked about was about the council, Life Science Sector Skill Development Council. We will quickly go through that. We will talk about the industry partner, Prerna Bioinnovation Research Center, and we'll cover the introductory part of drug discovery development, and if time permits, even the alternatives to animal experimentation. So, this is the course content. As you can see from that, all the topics which we have mentioned in the course content, I think most of them are covered. Only remaining ones are structure-based drug designing, either de novo or rational drug designing, and Qs are computer-aided drug designing. This we will be covering in classes to come. We talked about target selection, validation. I will again cover this in subsequent class from the point of role of chemistry in target selection, especially, and what is the role of biotechnology or a microbiology when we do the initial screening or a biological assays. We will cover that. And in vitro screening, we have talked about DMPK studies. We have talked in brief. We have talked about efficacy studies, but few specific models in a subsequent class. I will cover that. We have talked of safety toxicology studies, clinical trials. We have covered as a topic, but in detail we haven't discussed. So we'll try to cover that. And high throughput also in bits and pieces we have covered, but we'll try to touch base on the entire high throughput screening in drug discovery again tomorrow. Okay, so that's about the course what we started on 21st of or 22nd of December, or 21st of December. We started with introduction to Life Science Sector Skill Development Council, right? So life sciences basically, as you all know by now, it's a non-profit, non-statutory certification body which is set up under the mandate of Ministry of Skill Development and Entrepreneurship. Okay, it comes under NSDC, National Skill Development Corporation. Okay, it is based out in Delhi, and the life science sector itself is very important. As all of you are from same field, you should know that India is the third largest exporter of the vaccines. As in today's COVID scenario, we know that not many countries have any breakthroughs in terms of vaccine for COVID, but India does have, and we have Serum Institute, we have Bharat Biotech, and we have Zydus Cadila, who have breakthroughs even in vaccine for COVID. So we are the third largest exporter of vaccines. Okay, and from a life science perspective, India definitely is an important country. And in terms of council, what is the job? The major objective of the council is to fill the skill gap, which is there between academia and industry. So we try to bring them together, try to align them so that industry-ready candidates can be made available from academia for industry. Okay, so as I mentioned, it is a national level skill certification and apprenticeship body through NSDC. Okay, LSSDC has developed around 60 job roles, okay, which are covering more than 80% of the job requirement across country in pharmaceutical industry, in life science sector. Okay, and more than 45,000 employees across life science sector, pharma especially, we have certified so far through Life Science Council. In figures, we talked about more than 300 training partners are there with the council, more than 600 industry partners, and as I mentioned, more than 60 qualification packs are there, and more than 11,000, that number has now become, as on date while I'm talking, 15,000 apprentices have already been registered in different pharma sector. Placements, we have assisted more than 20,000 students to get placed in life sciences sector, and in academia as well as in industry, we have certified more than 400 trainers for LSSDC related job roles. So when I say Life Science Sector Skill Council, so what all things comes within the purview of council is pharmaceuticals, you have biopharmaceuticals and contract research. So under pharmaceuticals, all the formulation units, right? All the export-oriented formulation companies, API manufacturers, contract manufacturers, and nutraceutical products. Everything comes within the purview of life science sector. Under biopharmaceutics, we have biopharmaceutical, and in contract research, we have got both preclinical as well as clinical research within the purview of life sciences. Then we also talked, as I already mentioned, that by volume, India is the third largest pharmaceutical industry in the world. That's why it's a very important sector. So all of you students should be happy about that. The field, the sector which you belong to, has a huge scope. Okay, so your job or from your career perspective, you are in the right field. In terms of close alignment with the government, LSSDC, which is License Sector Skill Development Council, we are working very closely with CDSCO, right? Central Drug Standard Control Organization, Department of Pharmaceuticals, Ministry of Chemicals and Pharmaceuticals, and DBT, which is Department of Biotechnology. There are a lot of industry associations. I would say this industry associations, which I'm going to name, cover more than 90% of the life science sector within the state of within the India, which includes bulk drug manufacturers and association, Federation of Pharma Entrepreneurs, Indian Drug Manufacturers Association, Organization of Pharmaceutical Producers of India, Association of Biotechnology-Led Enterprises, Pharmaxil, and IPA, which is Indian Pharmaceutical Alliance. So for students, as we discussed in the first class, in terms of their employment snapshot within the sector, so you see more than 48% job lies for manufacturing. Okay, more than 10% for R&D, 24% for sales, and 5% for quality assurance, and then 4% for logistics and other department. So even today, more than 50% job role opportunities lies with manufacturing sector within the life sciences. From qualification perspective, all the way from a 12th pass or a 10th pass to graduate to postgraduate to PhDs or ITIs or diploma engineers, all of them have a scope. And if you see the distribution, around 5 to 8% are always the PhDs and masters students involvement. From graduation point of view, more than 25% employability is for the graduates in life science sector, and for ITS and diploma, again same amount of employability or employment opportunities are there, up to 25%. And 12th and below 12th, lowly qualified people or ITIs or B Farm, they also not B Farm, I would say diploma, they also constitute around 20 to 25% of the qualification requirement within the industry. So in nutshell, no matter where you fall in this table, you have a role to play in life science sector if you are interested in doing job or if you are looking for an employment. About apprenticeships program, students have already asked a few questions about that, so I would like to clarify that there is a scheme run by the Government of India called as National Apprenticeship Promotion Scheme. So under that, you can register yourself as a student, and then after registering, you will be available. I mean, you have to fill in the details about your own detail, your name, address, phone number, Aadhaar card, educational qualification, your address proof, all those documents. Once you upload, you will get one number, that is an enrollment number, and then you will be accessible by employers pan India. They can now anytime, if they look for apprentice and they search for a particular qualification, your name will automatically appear to them. So you can also yourself search for open apprenticeship opportunities in different companies and apply for that. And through council, license council also, we try and help you get aligned to the industry where there is a scope for you to join as apprentices. So that's about apprenticeship program. And again, you get a stipend paid during the apprenticeship period, and after the stipend, based on your performance, companies can take you on their payroll. So till date, as I was mentioning, more than 15,000 apprenticeship trainees have been already aligned within life science sector by LSSDC, that is by the council. And what are the job roles for apprenticeship? Like All 60 are available, but majorly quality assurance, manufacturing assistant, maintenance assistant, machine operator, production chemist, bioprocess engineer, store assistant, packaging assistant. These are all the very common job roles under which you can be absorbed in industry as apprentices. Then council also runs e-certification programs online, like right now the course which you are doing through APSSDC. So we collaborate with state corporations, and then we run such programs. So right now we are running 3 programs on production, pharma production, medical representative, and quality analytics. One of the important online program which we run under our initiative called ePathshala. When you go to the site, you can look for enroll. Under that, you can see ePathshala. These are 1 week programs only, where we try to have a snapshot of entire drug discovery development within 1 week. We are right now doing the course for 15 days. There we try to cover whatever best can be covered within 1 week time, and then you have to do an assessment and you get a certification. Same way, analytical technologies in drug discovery and development, that is also one of the online program which we are running in ePathshala. There are 5 centers of excellence from Life Science Sector Skill Development Council, namely Kerala, Microlabs, Macloids, 2 centers of Macloids, especially in Himachal and in Gujarat, and Swarna Bio Trust, which is in Shamshabad Mandal in Telangana. Okay. So this is the ePathshala brochure. As you can see, ePathshala 1 week accelerated webinar courses. So you can, if at all after this course is completed, if you are interested, you can register yourself and then you can enroll yourself for these courses. So very quickly, let's take few things on the knowledge partner Prerna Bio Innovation. Again, it's a contract research organization. So basically, this is an organization who does this drug discovery development on a contractual basis. So the entire course is about drug discovery development. So what we have done what we have covered in last 10 days, most of those activities are being carried out at this organization because it's a contract research organization. So like that, see, there are many CROs across country. If you are aligned towards research, then you can be absorbed in any of these CROs also. So all the fields which we have talked about, bioanalytical, microbiology, biotechnology, pre-formulation, chemistry, drug metabolism, pharmacokinetics, toxicology, all these services are provided by PBR. This is the virtual snapshot of the lab, how it looks like. There is a state-of-art animal facility which is registered with CPCSCA, and this is how it looks like. Few more snapshot, and one of the success story with the lab, because we are talking about drug discovery development, just wanted to share this as an example. Like this is a novel enzyme mixture which was launched by Karyotica Biologicals, an Hyderabad-based biotech company, in collaboration with PBR, and Prerna Bio was responsible for doing all the clinical trials, field trials for this particular product. So as you can see, it is a mixture of enzymes. Basically, the mechanism is it improves the digestion, and just by improving the digestion, you will be surprised to see that the efficiency is increased immensely. So you see, just by improving the digestion in the milking animals, you can actually increase their efficiency in terms of increased milk yield and increased fat percentage. So that was the product launched back in 2016. These are the few pictures from the official launch at a government function, and it was duly covered in the media because it is purely natural, and just by improving digestion, you can increase the efficiency of the milking animals. More recently, PBR has come up with this portable disinfection box in the period in this current COVID pandemic crisis situation, which is again working on the principle of UVC, that is UVC light at a frequency of 254 nanometer, and at that light it is completely germicidal. It can kill virus, bacteria, fungi, or any other microbes. So it is completely germicidal, and if you expose any articles which includes your currency, your coins, wallet, phone, watch, or specs, or anything you keep in that for 8 minutes, they are completely sterile, they're completely virus free. So it's high, it's very portable unit. Why I'm sharing is because drug discovery development is all about novelty. Again, it's not a new discovery because this is a very old stuff. As most of your microbiology and biotech students will know that we have been using laminar flow, right, for this many years. Where again we use UV light to make the area sterile. Correct. It's only this is a special light. It is a UVC light. There are three kinds of light: UVC, UVA, B, and C. This is UVC light, which is at a specific wavelength of 250 nanometer. Okay, so slightly different than the UV light which you find in laminar flow. And PBR is also into too much of industrial training. They help students doing their dissertation work, novel research topics selection, help in their thesis work, and they do many of the summer training. So few snapshots from that. And even right now, one of the batch is going on for the industrial training. So where you get hands-on on all the areas which we have talked about, say bioanalytical, animal health facility, pharmacology, toxicology, even microbiology, biotechnology. So all those trainings are being imparted. So let's jump directly into drug discovery development, and I'll try to cover as many topics as possible as a revision, so that tomorrow again we can take a new topic. So as we have, drug discovery development is a right because uncertainty, drug really any stage. So it's really interesting to see the whole cycle of drug discovery and development. So I always say that it is full of fun because you don't know what's going to happen next, but it is very thrilling experience. So it keeps everyone really motivated. Okay, so that's why it is compared to a roller coaster ride. Also, we have now, all of you know, when you take an injection, even if you have a sore throat, how the drug is going to reach your throat. So this is just a cartoon. I think in the last 10 days we have learned in detail about absorption, distribution, metabolism, excretion. Gone are the days when discovery used to happen by chance. But now you can really do a very organized, planned discovery of novel drugs or novel molecules for the treatment of dreadful diseases. COVID may be one of those. Okay. In earlier scenario, of course, discovery used to happen by chance. Like penicillin is the biggest example. It was never meant to be a plant discovery. It was a contamination found in the plates, and later on it was realized that it's actually penicillin. It made it into penicillin. So, like that, many chance discovery used to happen. But with advancement in our knowledge, now I think we can plan discovery. Again, one more cartoon we shared, and very important. And I hope today when I share this, you will all get the perspective of it, because last 10 days we have been talking about different phases of drug discovery and development. So patient is saying that I stopped taking medicine because I prefer the original disease to the side effects, right? And that's the hard reality. That even though we do 10 to 15 years of research, so much of investment, so much of detailed study on mechanisms of how a drug will act, about the potential adverse effects, still when drug is approved by regulatory agency and when it is launched in the market, you never know. There could be some adverse side effects coming out which can show up in the clinics. So that's the hard reality, but that's what you have to deal with when you're working in drug discovery and development. So we talked about the whole process. Now I can go very quickly because all of you have learned this different phases. So early drug discovery, then there is a preclinical development, clinical development, and then the drug gets approved by FDA. Long process, takes around 10 to 15 years. Very costly because more than $2 billion of investment, and chances of success 1 in 30,000, which is 0.003%. Why it is 0.003%? Because from 30,000 compounds which might be synthesized by chemist or a medicinal chemist, hardly 2,000 will reach preclinical development. Very few will enter in phase 1. Even fewer molecule goes to phase 2. By the time it is in phase 3, less than 10 molecules, and only one will give you the return on investment. That's why 1 by 30,000, so 0.003% of success rate. In terms of different phases, now all of you know very well what we've been doing. What we have discussed now. So we talk about early drug discovery. So target identification is one of the very important and the first step in drug discovery, because first and foremost you need to come with a target. So you have to decide an unmet medical need or a disease which you want to work upon. Once you identify that, then you have to find out the target, okay, which is essentially, now all of you know, is going to be a molecular target, either a receptor or an ion channel, or it could be any of the, what do you call, enzymes. So in nutshell, is going to be a molecular target, and only identification of target is not going to suffice your purpose. You need to validate that target, right? Once you validate the target, then we can go for hit identification. You can run through a software, or you can do a biological assays, but from those hit you can really find a chemically diverse molecules or scaffolds which can show good potency or good binding to your molecular target, and those hits or leads then can further go into lead optimization. In preclinical, we do all kind of in vitro assays, as we have discussed about ADME assays, solubility, octanol solubility, all those assays. We do proof of concept study in animals as well as in vitro setup. We do formulation studies, and finally we do IND enabling studies, and we make an IND application. In clinical development, phase 1, 2, 3. Phase 1 always happens in healthy individual. Phase 2 and 3 in patient population. Phase 2 is in hundreds up to 500 patient population, and from 1000 to 5000 you can consider it as phase 3 clinical trials. And along with that, all the dose escalation, safety, efficacy, everything goes in. Finally, it goes for an FDA review, and once they approve, your drug is launched into the market. And once it is launched, we do a survey to look for any potential side effects or adverse effects because of the drug. Then that is called post-marketing monitoring or surveillance. So the summary is, your project starts with a target identification, followed by validation, hit identification, lead optimization, testing in vivo rodent species under preclinical development, going for IND approval. Once you get an IND nod for first in human, do your clinical trial, phase 1, 2, 3, and then apply for registration, ND application with FDA. Once they approve, it basically can be launched into the market for the treatment of disease. So what are the fields involved? Now all of you would have clearly understood whichever field you belong to, maybe pharmacy, maybe biotechnology, maybe microbiology, chemistry, or where do you see yourself, zoology, veterinary. So when target selection validation, there's a role of genomics. Your understanding on genomics, proteomics, bioinformatics really helps, right? When you are in a lead discovery, a lot of structural modification needs to be done. So chemistry comes into the play. Your pharmacology background comes into the play in doing the assay development. Your analytical background helps in doing high throughput screening. So different fields have a role in different. Finally, if you have experience in clinical research, or if you have studied clinical research, you can be a part of clinical phase, clinical trials, right? If you are from a zoology or a veterinary background or a pharmacology background, you can be a part in vivo studies, which happens in disease models or in healthy individuals for studying pharmacodynamics and pharmacokinetics. For biologics, we talked about it, the difference. Small molecules are very simple, small molecular weight, chemically synthesized. They are called chemical drugs. And biologics are generally complex because they're too big, large, right? Like I've taken an example of a monoclonal antibody. Apart from their size, even the structure, they're simple structures compared to biologics. The chemically synthesized small molecule have simple structures. Biologics, you can imagine, it's not about the size, even the post-translational modifications are there. So everything from monoclonal antibody, recombinant proteins, cytokines, enzymes, all this thing qualifies to be a biologics. The process As chemically synthesized starts from some raw material, it has to be a chemical or a reagent. Whereas in biologics, it starts with a living cell, right? You generally start with transfection of DNA in a host cell. Then you go for a cell line selection. Further, you go for a cell bank preparation, and then you can go for scale manufacturing, upstream, downstream, and then once you have the sufficient cell cultures isolated and purified, you go for characterization, you do stability studies, and then it converts into a formulation, then refrigeration, storage, and then finally patient gets it as a treatment. So the basic difference, always remember, small molecules, right? They are chemical medicines are small molecules or chemically synthesized drugs. Once you have your reactions validated, almost every time you can reproduce it with same amount of yield for a chemist. But when you are working with biologics, since you work with a living cell, since you are trying to do something at a molecular level, some changes in the protein or some genetic blueprint you're trying to work through, any change in your process, in the entire process, will lead to difference in your final product. So biologics are little complex molecule to work with, because any change in our entire process will lead to change in quality, change in yield of your final biologics product. So that's the difference between biologics and chemically synthesized drugs. When you go for an IND submission of a biologics, again, the regulatory agency actually looks that from a similar point of view as they look for a small molecule. Basically, they ask for is pharmacodynamics, because biologics, they would like to understand the entire biology of that. What is the activity? What is the mechanism? And what is the potency you are proposing? They would also like to understand the pharmacokinetics. Now you know the distribution, elimination. I'm sure all of you have understood by this that what is area under the curve, what is concentration maximum, volume of distribution, and half-life. Correct? And of course, they would like to understand if there are any toxicity potential in your biologics or anti-cancer biologics. So typically in a non-clinical scenario, this is the data which regulatory agency looks for when you file an IND for a biologics. So drug discovery development funnel, you start with thousands of molecules and you end up having only one successful molecule which is safe and which is efficacious and which is successfully launched in the market. Total time taken, close to 15 years. Investment, more than $2 to $3 billion. Yeah. Drug development, so what we understood, so it is very costly affair. But for you students, it is good. Since it's a costly affair, since there is a huge investment into R&Ds done by most of the pharmaceutical pharma giants, you have great scope in this field, right? So from a cost perspective, of course, it's a costly affair. So when somebody says that a pill or a capsule is costing too much, now you should know, because a lot of cost goes into preclinical phase, a lot of cost goes into clinical trials, and even after the drug is launched, close to $100 million are spent on that. That's why that each capsule or each tablet costs you much. At the same time, this cost also is more because there is a huge investment on compounds which eventually fail, which cannot make it, because from starting onwards you can't have a compound which is going to be successfully launched in the market. You start with 30,000 compounds, example, and you end up having only one molecule. So your investment goes into those 29,999 compounds also, which could not make it into market. So that attrition, that failure also cost you. That's why the cost of that one successful compound or a drug candidate or a novel medicine is more. Okay. So let's talk about now failure. I'm talking about many compounds fail at different phases. These are the few reasons. As I already mentioned, this is an old slide, but let me first explain this. More than 40% reason is poor pharmacokinetics, for which a drug can fail in drug discovery and development. More than 22% reason, more than 20% and more than 30% for lack of efficacy and toxicity. So these are the major reasons. As I'm saying, it's an old slide. Now this 41%, we have come down to hardly 10% or less than 10%. Why? Because our understanding of ADME properties has increased immensely, and secondly, because of the technological advancement, it is now possible that you can really predict the poor PK properties very early, and you can actually fix those issues. All right. When a drug is in a preclinical phase or in a different clinical phase, this is just a survey where they have seen what are the chances for the reasons, maximum percentage reason drug can fail. So always remember, phase 2 and 3, it is because of lack of efficacy. Phase 1 because of clinical safety, and in preclinical, by and far mostly drug fails because of toxicology reasons. Just an example from Pfizer, millions of molecules you take, you screen through preclinical development, then get into in vitro, in vivo assays, efficacy studies, toxicology assays, studies, clinical trials, and by the time you are ready to launch your product, it's only 1 or 2 products. You see, so we say it's a very long, costly process. Even after doing this much of research, this many years of detailed research about every single aspect, be it pharmacology, pharmacokinetics, or toxicology, or formulation aspects of the compound, still, once the compound is successfully launched, there are chances that drug needs to be withdrawn from the market. Why? So this is the list of the drugs which have been withdrawn in different years in the origin country. And the reasons, if you see, by and large, majorly because of hepatotoxicity, heart attack, lack of efficacy, no improvement in clinical benefit, risk to heart valve, liver toxicity. You see, these are all the major reasons for failure. So if I had to just broadly classify this into 2 categories, so drugs are failing in clinics after this many years of research and successful launch is either because of lack of efficacy or because of side effects, toxicological effects or adverse effects, right? So this is the whole summary which we already talked about. Then we also covered this, that actually what are the attributes you look for, or why actually it takes this many what do you call it, this much of time and cost, because you have to qualify your drug candidate, your novel drug candidate, for all these parameters, right? We want it to be targeted. If it is not specifically targeting, it is not selective, it may impact some other target, molecular target, and it may have some adverse effect related to that additional target, which is not our intention. So it has to be targeted, the drug has to be selective. Of course, it has to be potent. There was a time when drug used to be taken further into drug discovery development, even if the potency is in mgs or micromolars. Now we try and have as potent drug as possible at the lowest possible concentration, even in say nanomolars or picomolars. Correct? It should be physically stable. Now you know what is the reason it should be soluble. If not, then a lot of effort goes into improving the solubility. It should be, of course, permeable, only then it can cross the intestinal barrier. It should be metabolically stable. Again, it's debatable because based on the target you're working on, it can be decided. It should be non-inducing to cytochrome P450 enzymes of your liver as well as of your intestine, right? It should be reversible. Again, debatable based on the target. Duration, again, it depends on the target. If it is antibiotics, we want the duration to be less, shorter half-lives. If it is for diabetes, if it is for any other neurological disorder, we want the half-life to be longer so that frequency of dosing can be reduced. We don't want any toxicity. So now you know it shouldn't be teratogenic, it should not be mutagenic. There should be a possibility to do a patent of that. And finally, you should be able to scale up. No matter how potent, how safe, how optimized compound you have, if you cannot scale up the manufacturing, then also the drug can fail. So these are the reasons why it takes so long for drug discovery and development, and why it's a very costlier affair. Okay. So, in nutshell, once you select the target, once we really identify the target and validate that, you should have a biological assay in place. Okay. So what you do with that biological assay is we will generally screen the molecules. It can be a cell-based or it can be an enzyme-based assay. And once you do that, you would also like to check the efficacy in a disease model. So that can be done through in vivo testing. Correct. And while I was talking about in silico screening, so there are computer-based softwares which are available. So each companies have their own compound libraries from different projects, from different disease projects. So what they do is they will run all these in-house libraries against the target which you selected. And if you find some hits, some of the structures, if they are trying to, if they're fitting well within the molecular 3D structure of your target, then that becomes your first choice with which you would like to start your program. So that's one way of doing screening, which is called in silico screening. And then the other ways, you already have a published literature. You can take a structure from that, and then based on that, then there are natural products from where you can do. And we'll be talking about rational drug designing in the next class, I hope. And you can do a proof of concept study. So now I think this whole process, which was really something new for all of you in the first class, I hope now you are at least getting the meaning of it and the application, that where it has to be done, how it has to be done, why it has to be done, what is the application of all this thing. Okay. So then we talked of alternatives to animal experimentation. So do we really need animal experimentation? I don't want to spend much time on it because all of you have really understood animal experimentation is must. You can reduce the usage of animal, but you can never completely replace usage of animal in research. You of course need a drug experimental design, because there is a big difference to counteract the variability. This particular picture tells you, right, you cannot really give them the same task. It's like different animals have different surface area, different physiology, different anatomy. So there's a huge variability. So you should have an experimental design in place to reduce this variability. Okay. So in alternatives to animal experimentation, we talked about hepatocytes and enterocytes, right? So if you don't want to really go for animal experiments at the first place, in fact, when I started my career, I must say that almost 20 years ago, I used to get compounds from chemist, and every single time I was supposed to inject or dose into animals, live animals, and then look for PK properties. And 9 out of 10 times, the issue was lack of viability. There were no concentrations found in the plasma or the blood samples collected from those animals after administration of the dose of the drug. And what was the reason? The reason was we used to do reverse pharmacology, and as you now know, drug might have degraded in stomach, it might have degraded in acidic pH or in neutral pH, it may be a substrate to transporters, somehow it may not have crossed the intestinal barrier, it may be a substrate to enterocytes metabolism, first pass effect in intestine, or it might have been completely metabolized in liver, or after coming in contact with esterases and hydrolases in the blood, it would have degraded. So there could be plenty of reasons why I don't see concentration of my drug in samples. So then we used to do all these in vitro assays, correct? So that's the reason I say that now there is a paradigm shift. Most of the companies, they start by doing in silico assays, whatever they can use, they can do using computers, and after doing that, they come back and do in vitro studies, and after in vitro they do in vivo studies. So in vitro, we have talked in detail about two tools, two models, cell-based model. One is liver cells, which is hepatocytes. Another is intestinal cells, which is enterocytes. Correct? So as we understood the fate of a drug now very well clearly, if it is given orally, it goes to your stomach, intestine gets absorbed, reaches the circulation, through portal vein it is delivered to the liver, from there back to the circulation, and from systemic circulation it goes to the different organs. So the absorption phase, the role of pharmaceutics comes into the play. After absorption, once it is in the blood, the unbound fraction, the free fraction, is available for eliciting the pharmacological action. So that is called free fraction. So free fraction can can elicit the required action at a receptor level or at a molecular target level, or it can go to the tissues and can bind to the proteins in the tissues. That's called non-specific binding. Within the blood, it goes to liver, and in liver, if it gets metabolized, then the metabolites are highly polar and it gets excreted, or as such drug can be excreted through renal excretion. So this is the fate of a drug after following oral administration. If it is any other systemic circulation, then only the absorption phase goes away. Directly drug reaches to the systemic circulation if it is parenteral intravenous route, right? So what all body is doing to the drug, body is absorbing, distributing, metabolizing and eliminating the drug. So that is called pharmacokinetics. And what drug is doing to the body, which is it is eliciting the pharmacological action, that is called pharmacodynamics. So drug metabolism, we talked about it, that drug has to pass through intestine. So intestine, in enterocytes, there are expression of cytochrome P450 enzymes. So metabolism can happen in intestine. From portal vein, it gets, suppose it passes through that, through gut wall, it gets absorbed through portal vein, it reaches liver, and then in liver again cytochrome P450 enzymes are expressed in abundantly, and metabolism can happen even in liver. Most abundant isozyme, cytochrome P450 enzyme, which is expressed. So as I mentioned, more than 50% or 75% of the drugs available in the market are metabolized by single isozyme, which is cytochrome P450 3A4. So CYP3A4 is abundantly expressed in intestine, and same 3A4 is expressed in liver. So now you see that's the reason we should do metabolism studies in intestine as well as in liver. So we talked in detail about hepatocytes. They are by far considered as gold standards for in vitro hepatic metabolism. Okay, and why they are so popular? Because they got intact plasma membrane, they got complete uninterrupted enzymes at a physiological conditions, and they do express the transporter proteins, right? Both kind of transporters, efflux as well as uptake transporters, mainly PGP that is expressed. So that's why hepatocytes are considered as gold standard for in vitro metabolism. So what is the advantage of using these hepatocytes? So as you may understand, we need to really cryopreserve the hepatocytes so that they can be transported, they can be stored, they can be reused. So what is the basic advantage of doing this cryopreservation? So the advantage is the experiments can be scheduled because you have a cryopreserved lot in your facility maintained. You know you can really plan your experiment ahead of time. You want to repeat your experiment. Suppose you got 3 vials of hepatocytes, 2 of them still remains in a cryopreserved form. One you can take it out, complete your assay. Remaining you can repeat the day after or whenever you want. So these are all the advantages. Your experiment can be scheduled. You can repeat the experiment, even pre-characterize, as I'll be sharing the characterization sheet kind of thing. So all those things can be possible just because of cryopreservation. As I mentioned, there are different lots. I've taken this example from one of the vendor companies, which is In Vitro Admet Labs, IVL. They are the supplier of hepatocytes, one of the suppliers for hepatocytes and enterocytes in India. So based on the age, gender, race, and specific yield information and viability is always available for any lot of hepatocytes which you procure from market. In general, the yield is somewhere in the range of 6 to 7 million cells per vial, and viability is always more than 90% for hepatocytes. Then there is something called as platable hepatocytes. So we have talked about hepatocytes. they can be in a suspension form, they can be in a platable form. As you may understand, if it is in a platable form, it is a high throughput fashion. You can do it in a 96 well plate also. So that's the advantage. And what happens is platable can be cultured for a longer duration. Correct. So if it is a suspension, the half life is hardly 6 hours. So you can do small studies like drug uptake, metabolic stability, metabolic profiling, which can be completed within few hours. Then suspension is a better choice. If you want to do long-term studies like long-term metabolism or in vitro hepatotoxicity, where you need longer incubations than hours or days, then plated hepatocytes are the better option. This is how, as we have shared, the cells hepatocytes looks like across species. Even the cytochrome P450 enzyme expression can be studied in each of these cells. As you may see, omeprazole, phenacetin, and rifampicin. These are the known substrates. We have used that to study the induction of CYP1A2, like 2B6, 3A4. These are all the isozymes, major isozymes of cytochrome P450, which are responsible for metabolism of the drug. So if you are trying to do an in vitro assay and if you want to see the expression of these enzymes, you can very well do that. So more than 90% viability, 6 to 7 million per vial, nearly 100% confluent, and major isozymes are you can take down. It is 1A2, 2B6, 3A4, 2D6, 2C19. So these are all the major cytochrome P450 enzymes. And of course, there is an expression of transporter proteins which could be uptake on efflux. And over 7 days, you can do the culture. So that's an advantage of having platable lots. You can use from different species basically. Any vendor you use, as I'm saying, I'll just for the example, I'll use IVAL, but you can use other vendors too. But this is most prominent vendor whose hepatocytes are really viable and they show very good yield. And they're very rugged to handle. I mean, they pretty much work most of the time. So that's why most of the companies are using from IVL. That's why I've taken example of IVL. But when you're doing an in vitro screening of your compounds, you can always generate data across species, so that when you try to extrapolate the data into the live species, in vivo, higher species, the extrapolation will be more accurate. So all the lots like in human, mouse, rat, beagle, cynomolgus monkey and rhesus monkey, these are all the platable hepatocytes readily commercially available. There's something called as quick freeze. It's nothing but they are cryopreserved human hepatocytes, thawed, mixed well, pulled, and then again refreezed. So still they can retain their activity. That's the beauty of this. It's a patented technology. The advantage is you can have pulled human hepatocytes, because as you know, we try to control the variability. If you are using from one lot, okay, it doesn't really cover the variability. So we want different lots. So rather than repeating the experiment in different lot, we try to pool the lot, and it is available as a pooled human hepatocyte. So whatever data you get from that, at least you know that the variability is taken care of to minimize the individual differences. Okay. So where is the application of multiple donor? Which assays? So when you do a metabolic stability, profiling, reaction phenotyping, P450 inhibition or induction assays or hepatotoxicity, multiple donor hepatocyte is a better choice. When we talked of enterocytes, why should we study enterocytes metabolism? Metabolism in enterocytes because cytochrome P450 3A4 is evidently expressed even in intestine. So why enterocytes? Because friends, as we have discussed this in past, the first organ and one of the first organ which is going to see your drug after oral administration or oral dosage form is intestine. So intestine is going to see your drug first, and your drug needs to permeate through that. Only then it can be absorbed, it can reach the vasculature through portal vein, then it can go to liver. So the first initial organ to see your drug is enterocytes, and there you see expression of cytochrome P450 enzymes. So that's why it is very important if you are looking for oral viability enteric metabolism could be one of the limiting factor. So it's better to study metabolism in enterocyte before actually testing your drug in animals, in a live animals. As I already mentioned, enterocytes are not available right now, but I think now it is available as cryopreserved human intestinal mucosa. So when you see this is how they look like. They are small in size, compactly packed. Compared to hepatocytes, they are definitely smaller, and they are mainly single cells. In terms of protein content, they have lesser protein compared to hepatocytes. Hepatocytes will have somewhere around 1 to 1.5 mg per million cells, whereas human enterocytes will have hardly 0.25 mg only. Okay. Yield and viability. So as we already have talked about, the viability is definitely less. Anything above 75% viability you get in enterocyte, that's you are better off. I mean, you are it's better, it's good. The yield is also somewhere around 2% only. I mean to say, 2 million cells per vial compared to hepatocytes where you have up to 6 to 7 million per vial. In terms of cryopreservation, as I said, successful isolation cryopreservation, but viability less than hepatocytes, but still more than 75% in yield of 1 to 3. Same information, detailed information of the donors are always available with all any vendor you use. How do you really differentiate an enterocyte from a hepatocytes? So there are marker gene expressions you can look for. In enterocytes, there is an expression of sucrose isomaltase and maltase glucoamylase, whereas in hepatocytes, it is albumin. So if you see, these are cell-specific markers. If you have expression, if you have intestine, then you should have sucrose isomaltose and MAGM expressed in that. And if it is hepatocytes, it won't express this too, but instead it will express albumin. Okay. I already mentioned this that major enzyme responsible for metabolism of majority of the marketed drug is cytochrome P450 3A4. Same 3A4 is expressed in intestine and in liver. In intestine, out of all the CYP expression, 82% is only CYP3A4. So you see it is abundant in intestine. Whereas in liver, 40%, still it is in majority, but it's 40% expression is of 3A4. So that's why apart from liver, we should study intestine also. Apart from 3A4, if we talk about other isozymes, there are few specific isozymes which are only expressed in enterocytes. So now you will understand if at all your drug has a potential to get metabolized by those isoforms, say 2S1, then intestine enterocyte is a better model than hepatocytes. Correct? So like that, there are a few others like CYP2J2, CYP4F12. They are all expressed in intestine. Okay, in hepatocyte, most of them are expressed, but I think 2S1 is almost negligible in hepatocytes. So for 2S1, by all means, enterocyte is a better model. This is published in the literature as well that 2S1 is specific to enterocytes. Okay, so we have talked about enterocytes. Let's talk about enterocytes application part. So enteric metabolism. To study enteric metabolism, enterocyte is one of the important tool. You can study drug-drug interaction. Yeah, if more than one drug is taken orally, then whatever drug-drug interaction can happen, that can be studied in intestine. You already take food, right, during the day or a night time, and that food will also be there in the intestine. So you can study food and drug interaction also. And if there is any enteric toxicity potential of your drug, that can also be studied using enterocyte. So that's the application of using enterocytes. Then we talked of near perfect hepatocyte cryopreservation. So this is also commercially available. So this is near perfect. That's why it is called 999 elite group, elite hepatocytes. So it is nothing but more than 90% viability, more than 90% confluency, and more than 9 days of culture duration. So three nines are there. That's why it is called 999 elite cryopreserved human hepatocytes. Then there is some tool for you to use called as MatMax human hepatocytes. This is slightly different than normal hepatocytes, and we have understood why. Because MatMax is nothing but it is a permeabilized cofactor supplemented hepatocytes. So cofactor, more and more cofactor has been added to it. So now they are permeabilized. Let's see in this diagram. If you see an intact hepatocyte as an intact membrane here, we have made that porous. So that becomes a MatMax hepatocytes. So now by doing so, there are definitely few advantages. As you can see in terms of endoplasmic reticulum, cytosol, mitochondria, lysosome, all the parts of a cell, typical cell, plasma membrane, nucleus, MatMax is pretty much same as an intact hepatocytes, right? Of course, it is definitely better than microsomes and S9 fraction, which are generally used for first tool to study the metabolism, but this can definitely be used in place of hepatocytes if you want to make your process easy. How? Let's see. The reason is to handle an intact hepatocytes, as I mentioned, you need a cryopreservation, and the storage would be always in minus 170 degrees, which would be either in a liquid nitrogen. Whereas MatMax can be stored just like a microsome in a minus 80 deep freezer. So logistically, it becomes very easy. Just take it out, thaw, use. You want to keep it back, you can keep it, or you can utilize it. So you see, when you use an hepatocyte, you have to do centrifugation, microscopic examination, cell counting. We all those things we do, right? Whereas in MatMax, you don't have to do any of those things. Just simply take it out from the minus 80 freezer, thaw, and use, ready to use. Evidently, there is a cofactors in it. So the only downside is, since we have supplemented additional cofactors, it shows you higher metabolism. You got my point? It gives you higher metabolism potential. Again, at least sometimes when there is a very low metabolism, so the detection becomes an issue. So sometimes it goes undetected while you're using hepatocytes. But with using MatMax, even that low potential of a drug to get metabolized can be captured using MatMax. So no metabolism can go unnoticed in an in vitro assay. So now you see, sometimes I have discussed this also in previous classes, that many times in human, both the metabolism, efficacy or toxicity is different than what you see in other species. Sometimes we are not able to capture the metabolism correctly. Isozyme expression could be still the same, metabolic pathways could be same, but since it was a low metabolism, we could not capture that using hepatocytes, and that happened in human. So at least using this, that can be avoided. As I discussed this in detail, for different herbal supplements, we have checked the inhibition of enteric 3 or 4 activity, and good to know that green tea and all, they actually do not inhibit, but there are many herbal extracts which have potential to inhibit 3 or 4 activity, enteric 3 or 4 activity. Okay. So what is the application of in vitro enteric system? In enteric metabolic clearance and enteric drug-drug reaction. So what is the take-home message there? What we have been visiting again and again is animal hepatocytes. When you do a study, you can also do a comparative study in human hepatocytes. Then you can extrapolate that data for prediction of properties, drug properties in vivo in human. Correct? Also, you have human hepatocytes, you have animal hepatocytes. You can do a study in animal hepatic drug properties by in vivo dosing. So now you have in vivo data, you have animal data, intestinal mucosa data, and you have human mucosa data. All three together. Now your extrapolation for enteric properties of human prediction would be best. I mean, it would be improved. So that's the purpose of using enterocytes. So all these years, hepatocyte used to be prediction of hepatic drug properties in human is what we were interested. But with this knowledge improvement in enteric properties, we can even predict the human enteric drug properties by doing in vitro assays in animal tissues, especially intestinal mucosas, cryopreserved human intestinal mucosa or cryopreserved intestinal mucosa from different lab animal species. Okay, so that's what we last time in our first, I would say 3 to 4 classes. Okay, and then very quickly, I think we have time for another 20-25 minutes. So let me revise this also, so that I know I've gone little fast today, but as you can understand, I'm trying to cover our 10 days of course, 10 odd days of course into 1 or 2 classes. So we need to be little, it's just to refresh you, so that I don't want you to spend any additional extra time to study for the assessment which we are going to do next week. So I'm just touch basing the topics which we have covered. So you can simply appear for the assessment, because all these topics you already have learned, and just by revisiting the slides, this will again refresh in your mind. Okay. So when we talked of target identification and validation also, so the first step in the drug discovery, as we have seen, is to identify an unmet medical need, to identify the target, and to understand the molecular target responsible for disease, so that by doing intervention of that molecular target, you can really treat the disease. Okay, so this is what we have studied so far. In the mechanisms, once they are studied, the target majorly would be a receptor, ion channel, or a transporter, or an enzyme, or a signaling molecule. Correct? We do a lot of such studies, proof of concept studies, genetic models we use to really see the proof of concept. That is knock-in or knockout mice, which is available now. And then we can go for a biological assay for an automation to identify the lead from hits. And once you have the lead, you do selectivity, efficacy, tolerability, PK studies, and all those studies can be done to optimize your lead. And then the drug candidate successfully optimized as lead can be tested for its toxicology potential, safety potential, by doing safety studies. And then finally it goes to human, right? And then the drug approval. So target identification, validation, molecular target, right? A biological hypothesis, and a genetical model to demonstrate the proof. High throughput screening of your compound libraries in silico, look for the hits, then do the in vitro assay. That's for hit identification. For lead, medicinal chemistry effort for hits to leads, because sometimes hits you get, then medicinal chemists will have to really synthesize this molecule. Again, go for potency, selectivity. If there are any issues, go back to medicinal chemist. They'll do some structural modification. Again, synthesize. Again, go and look for potency and selectivity. Then do in vitro, look for liabilities. If there is any CYP inhibition or induction liabilities, then go for in vivo efficacy. And lead optimization, we already have talked about, detailed in vivo efficacy, in vivo PK, dose range finding. All this thing has to be done in optimization. Finally, we go to preclinical development, where now all of you are well versed with the term GLP. So we want GLP compliant toxicology and PK study data, safety data, formulation data, and then you are ready to go for pre-IND meeting, right? So target selection, don't want to repeat, already we have covered this. This slide also we have talked about. Majorly is going to be a molecular target, right? 9 out of 10 times. Enzyme, receptor, protein interaction, key enzyme, ion channels, nuclear receptors, signaling pathways. This could be your possible potential drug targets. Of course, it has to be of interest, unmet medical need. Okay, you can do a proof of concept studies by developing a biological model for that, by doing a knockout or a knock-in mice or rats. High throughput screening. Once you have the biological assay in place, either a cell-based or an enzyme-based, you can actually do the screening of the molecules in a high throughput fashion. Of course, hits. Once you start screening the hits, you'll end up having a lead molecule, right? But once you have the lead, practically chemist has to synthesize those leads, all the related analogs, and then come back to you for testing of potency as well as toxicology. Once that is being done, if it needs to be further improvised, then you go back to the chemist, he will again modify the structure, again synthesize, you again check for the potency, again look for the selectivity, and then take it further. Another way of doing it is using computer aided drug designing, intuitive drug designing on medicinal chemistry. So this CADD we will cover in next two classes, and then we go for in vitro ADME characterization of a molecule, and finally you do a proof of concept study now in a disease model, okay, so that your whole cycle is over for initial hit and lead identification. So you remember all this thing is happening in a non-GLP fashion, okay. Just an example how from an existing compound, active compound also leads can be found out. As you can see, the structure of neurotransmitter serotonin 5-hydroxytryptamine is a structure. If you just modify the structure on this hydroxyl end by adding SO2 and amine group to this, actually it has formed a drug called as sumatriptan, and now it improves the biological activity. It is an agonist of 5-HT, right? There is a scope you can go for patent. It improves the physicochemical properties of this, and the therapeutic index has improved. So this is also one way of doing drug discovery, where you go with an existing structure and then try to modify. But at the same time, please look for the patent litigation. I mean, not litigation, but patent opportunities also. Sometimes if it is already patented, then you should be really careful in using the same structure, because any analogs, related analogs, would have been also covered in a same patent of that molecule. So we don't want to infringe the patent taken by somebody, correct? For lead optimization, one of the important factors is we also want to see the scalability, correct? If you're able to manufacture in large scale, as I mentioned, otherwise, no matter how potent, how safe, how optimal a drug candidate you have come up with, if it cannot be scaled up, it's of no use, right? So in vitro potency, selectivity, in vivo efficacy, in vivo pharmacokinetic studies. After doing pharmacokinetic studies, you get parameters like half-life, Cmax, Tmax, volume of distribution, clearance, in vitro ADME characterization, mode of delivery. All this thing has to happen under lead optimization, correct? So we have seen target selection, high throughput screening. Finally, chemistry effort comes into the play. They also check for scalability. Meanwhile, we do in vitro selectivity, in vitro metabolism. Every time there is any issue, we go back to the synthetic chemist to modify the structure. Again, check the potency, come back to metabolism, animal efficacy study, in vitro tox. And finally, only few compounds will pass all these stages and can be taken further for first in human, and then they are taken for safety studies and then first in human. So this entire phase is called lead optimization. So now you will very well understand drug discovery and preclinical. So you have target selection to hit to lead optimization. Everything happens within this stage, and then drug enters into clinical trial. Then we also covered about INDs, investigational new drug application. So what is that? Basically a data package which you submit to FDA to allow the initiation of the clinical program. So that is called investigational new drug application, an IND application. So basically to present the data to justify that compound exhibit pharmacological activity to meet an unmet medical need, right? So they want to see the efficacy, they want to see the safety data, and they want to see the formulation data as well, especially in IND application. Within the purview of IND application, the information should be in three broad areas we have talked about. If it is a manufacturing, then manufacturing GMP, GLP and GCP for clinical protocols, and investigative information, animal data, toxicological data under GLP. We talked about the quality issue. What is basically GXP is, I just mentioned it because X can be replaced by M, L or C, and for good manufacturing practice, laboratory practices and clinical practices. So basically, quality control and quality assurance, these two things really helps you in forming the GXP criteria, right? So what are the IND enabling studies? If you have any molecule and if you feel it is reasonably safe and it is highly efficacious, highly potent molecule, you want to go further for human testing. What all studies are actually required? So IND enabling preclinical studies are efficacy, safety, general toxicology, genetic toxicology, pharmacokinetics, ADME, reproductive toxicology, carcinogenicity, and special studies. So IND enabling studies, basically you submit this to FDA, right? So pharmacology, of course, most important. You should have an in vivo animal model to demonstrate the efficacy. Okay. Safety, the major thing to remember here is core battery. So your safety data covering the three core battery, that is three systems: cardiovascular, respiratory, and central nervous system. Covering these three core battery tests should have been performed for under safety pharmacology. Toxicology, very important. Species selection is equally important. Dose selection, administration route, duration, and endpoint. This is the framework of any toxicology study. Species selection, as I already mentioned, preferably a rodent and a non-rodent, so mouse or rat, and dog or any primate. The thrust should be try and select the species which is metabolically relevant to the target species. So that way, chances of complication or failure of drug because of lack of efficacy or because of adverse effect in clinic would be less if you select clinically metabolically relevant species. Dose selection, always try and cover the maximum tolerable dose. Also, a dose level where you don't have any adverse effect. This too should be a part of it. So, in nutshell, you can cover low, mid, and high dose. You can take, right. Duration, bare minimum is 2 weeks for toxicology studies, and it depends on the final duration of your treatment in the target species or in the clinical trial. That will really define the duration of toxicity study in non-clinical species. Route of administration, again, it should be same as you anticipate the route of administration in human. Try to keep the same route. Yeah. If it is oral in human, oral in non-clinical species for doing toxicology. If it is intravascular, do intravascular, intravenous, do intravenous in non-clinical species also. So typically in a toxicity study, you look for all kind of daily basis clinical sign, morbidity, mortality, or any other change in their clinical behavior. On a weekly basis, take their body weights, functional observational battery covering all the three tests. Finally, when you sacrifice, before sacrifice, you can take the blood, do biochemistry, do hematology, and do clinical pathology, and do quantification, right, of your drug. You can collect tissues, and in tissues also you can quantify the drug, quantify the biomarkers, and close to 40 organs are collected after sacrifice of these animals following toxicity study, any toxicity study, and then you prepare histopathology and compare it against the control. So typically you'll have a control, low dose, mid dose, high dose, and couple of recovery groups. And recovery group always include the high dose recovery group, so that whatever data you get, if you get a positive data, you don't have to do other two doses at all. As I mentioned, you can do drug analysis, and most importantly under GLP, if it is falling, because for IND submission, the tox study has to be under GLP environment. So documentation is very important. As in yesterday's class, we have covered in detail about good laboratory practice, how it has to be done, how it has to be maintained. I would say that this documentation is very important for any toxicology study, IND enabling data package study. Okay. As I clearly mentioned, it has to be GLP compliant. Okay. So, in a general toxicity study, we have talked about species, we have talked about dose, we have talked about route of administration, we have talked about duration. Let's talk about endpoints. So, endpoints we already covered. It could be mortality, morbidity, clinical signs, body weight, temperature, activity, hematology, clinical chemistry, toxicokinetics, or pathology. Toxicokinetics is nothing but pharmacokinetics done at toxicology doses is called toxicokinetics. Generally done along with toxicology studies. All you need to do is you will have a separate satellite group where you will dose them and do the PK studies. And the dose should be same as your toxicology groups. Genetic toxicity. So core battery of genetic toxicity is in vitro assays like AIMS, in vitro mammalian chromosomal aberration assay, or in vivo micronucleus test. Correct? So first two you have to submit before you go for IND submission, and the last one before you go for permission for phase 2 clinical trials. Pharmacokinetic study data should be there as an IND enabling data. That is, you should have dosed the animals with your drug. You should have quantified those samples, and you should have data on parameters like area under the curve, concentration maximum, at the time maximum, half-life, clearance, and volume of distribution. So all these parameters, I'm glad that we have discussed this in detail in few classes prior, and all of you will now clearly understand the meaning of this PK parameters, which was not known to you initially, maybe on first 2 days when we talk about it. Reproductive tox, 3 types of study basically. We try to cover fertility and early embryonic, embryo fetal, and then peri and postnatal. So reproductive toxicology can also be under these 3 type of studies. Carcinogenicity, please remember any drug which you are trying to bring into market which is having a long-term administration, you need to continue the treatment for more than 6 months, then carcinogenicity study is must. If you want to do a pediatric drug research, I mean to say pediatric drug clinical trial, you need to have carcinogenicity study data when you go for permission, when you go to ask for permission with regulatory agency for first in human dose. You should have carcinogenicity data if your drug falls in pediatric category. Okay. Then we talked about the guidelines. So these are all CFR part 21, CFR part 3, 12, 14, 16, 58, 50, 56, available all on www.fda.gov. You should open that and read investigational new drug application, IND, what is the requirement, also NDA application and what is the requirement, about the orphan drugs, about GLP practices for non-clinical laboratory animals, about human subjects, institutional review board, drug labeling, everything is given in detail. In your free time, you can review if you want to gain more knowledge on these topics. Of course, there is a serendipity chance. We already have talked about, taken an example of penicillin, which was a discovery by chance. Same is the case with nitrous oxide, which was a discovery by chance. So like that, there in past many such discoveries have happened simply by chance, which were not intended. Efforts were not put in to really discover those chemicals, but they were discovered just by chance. So during the clinical trials, the entire summary, discovery screen and lead optimization, when you say, means in silico, in vitro, genotox, in vivo study, all these studies are happening during discovery and lead optimization. In preclinical, all kind of acute 28-day tox studies, in vitro assays, AIMS and chromosomal aberration, core battery, all this thing is happening during preclinical phase. During phase 1 clinical trials, while it is going on, you can do general toxicity studies of 3, 6 or 9 months toxicity study in vivo micronucleus. This data would be required to get a permission to do phase 2 clinical trial. And in phase 2, again, along with that, long-term chronic toxicity studies can continue, or reproductive tox studies can continue. And then before you actually go for phase 3, carcinogenicity studies should really continue, because it goes on for up to 2 years also. Correct? So these are all the different studies which run in parallel under these phases. Correct? Then we talked of very important decision gates. So after about almost 11 classes, I think all of you have understood, is your target validated, right? Whatever unmet medical need you have identified, whatever molecular target you have identified, is it really validated? That is your decision gate, number one. Are these leads, I mean, once you have validated, you go for hits. Are you able to identify some hits or leads out of it? That is another decision gate. Once you find a lead, not necessarily that highly potent scaffold or a structure which you have found is always a structure which you can work upon. It may have some inbuilt toxicity potential. It may have some inbuilt formulation issues or ADME issues, or it may have some manufacturing issues you can't scale up. So all these things are there. Then the question decision gate is it really possible to develop this lead? Correct. Same way, when you want to go to human, before that, do you really are you really convinced with the safety data from a non-clinical study that your compound is good to go to human? Correct. So there are so many decision gates at different phases of drug discovery and development, where you can really take decision, go no go, whether to take your drug further or to drop or fail your drug there itself. Because please remember, the more further you go in this path of drug discovery development, and if drug for some reason fails, for any reason, more would be your investment. So if at all drug has to fail, then please make sure that it fails very early. So we say fail early, fail cheap. Okay, not that we want drug to fail, but if at all drug has to fail, at least let them be cheap. It should not cost us more money, right? Then we talked about GLP. Of course, in yesterday's class we have detailed covered this, but GLP is nothing but a quality system. Now I think no need for me to spend any time on this, because all of you are very well versed with the entire GLP process. It's a quality system, and you want a framework where studies can be planned, performed, monitored, recorded, archived, and reported. So we want a framework in place of quality assurance or everything. I mean, within the purview of GLP, even your infrastructure, your facilities, your manpower, your instrumentation, your documentation, your in-house quality assurance, everything comes, or even the archival facility, everything comes within the purview of GLP. So what does GLP ensures? If your lab is or institute is a GLP certified lab, or it is compliance with principles of GLP, it generally means it ensures the quality and reliability of the data which is being generated. We have discussed this. I won't spend time on this in detail. We have talked about SOPs on each and every procedure which is happening in your facility should be covered with an SOP. Okay, so please do what is written and write what you do. That's it. That's the simplest definition of standard operating procedures. You should have all the relevant forms, formats in place. All the instruments should be calibrated, validated. You should have all the records in place, all the plans, future plans in place. Study plan. No study can happen without a signed study plan from a TFM. If it is not there, no study can take place. So study plan forms a very important component of any study which is done under GLP. There should be a reporting format. We have discussed that in detail. Entire documents, be it any SOP, protocol, or any document, there should be a document control. It should not be readily, freely available for everyone. There should be a process. Of course, it can be available for whosoever wants it, but there should be a process for a document control. And finally, most importantly, we say that you should have an archival in place, correct? If you have an archival, then all the hard copies of the documentation, soft copies, tissues, or the samples or slides, all this thing can be archived. Generally, going by the guideline, it says 5 years after the completion of the study, you have to archive till 5 years. But again, it also depends on the sponsor and the lab to mutually decide up till what time they want to store or archive the samples, archive the documents, raw data, actual wet samples, wet tissues, or slides. Okay. We have covered this in yesterday's class. In test facility management, the head of the institute of any organization, any lab, or a department, the head would be called as test facility management. Under him, there are three important components. The quality assurance should be under direct reporting to him or her, and archivist should be also a direct reporting to TFM of the facility, and each and every study should have a designated study director. And study director will be responsible for execution of the study. So from preparing the study plan to appointing all the study personnel to appointing the pathologist, everything lies within the purview of a study director job role, correct? And all the support facilities like formulation, analytical, bioanalytical scientist, biostatistician, all these support facilities also somehow falls under the kitty of study director. So, in nutshell, if I have to, after yesterday's class, I don't want to repeat the GLP because it's very, very recent for you guys, and all of you are very clear in your mind about GLP process. Let me just explain you the typical flow of a GLP process. Now that you understood the importance of GLP, you understood how GLP principles can be executed in your own lab or institute or department. So the process is, it starts with a study plan approval, correct? First and foremost, you can actually do a pre-study initiation meeting. We can definitely do a pre-study initiation meeting where, suppose we want to do a toxicology study, a 28-day tox study. First, you have to call for all the concerned people who are going to be the part of the study and discuss about the study. Okay, so that's called pre-study initiation meeting. Before you even actually start the study, you have to do this meeting. So we'll be checking the feasibility, we'll be checking the availability of the people, availability of the instruments, availability of the animal test items, availability of the test system. All those things we discuss in this meeting, and then the study director will prepare a study plan, and then it gets approved. Once it gets approved, since I said it's a toxicology study, you have to put up an animal request. So we go back to the animal house in charge, and in writing we have to give them a request for animal, and then they will give you an animal. You have to ask for of course, even if you don't ask, they will give you the health certificate. It gets filed as a raw data that your study, along with the study plan, it goes. So you have animal requisition, animal receipt form, you have the health certificate, then you can put up a request to formulation, requisition, all in documentation, not verbal. Okay. In formulation, scientist will prepare a formulation. He can further send it for formulation analysis. That report comes back to you. So now formulation you have formulation receipt, you have a certificate of analysis. Now you can actually do the experiment. The study personnel will do the dosing of the animals, and they start either in vitro or in vivo exposure, whatever you can start. And then the observation period, if you're doing a clinical observation, there should be a form that do you check the ophthalmological examination, any skin infection or infestation for different organ system, what all things you want to see clinically. It should be in a form of a format, so that somebody has to daily check that and sign it, right? Same way, all observation, like your feed intake form should be there, your body weight form should be there, your water intake form should be there. All those things should be there in a format. You simply cannot just take it down in a rough thing. You want it to be in a well documented manner. Finally, you have to send the samples for analysis at the end of it, or a pathologist to prepare a histopathology slides. Then they come back to you with the results, which again needs to be documented, and then generally a draft study report is being prepared. Okay, and that goes to quality assurance. This is not an external quality assurance. Your internal quality assurance unit, you go back to them, you ask them to audit the report, and they will actually audit the report. And since you would have intimated them during the pre-study initiation, they, in their schedule of a quality assurance unit, they would have already planned in study audit or in process audit or facility audit. So that report and this your report gets along with the report, and your final report is now in place, which has to be signed by the study director and finally by the test facility management. So QA can put up a letter, I mean a page certificate that the so and so study number and study title was done as per the principles of GLP. That means once you write, the QA write this certificate and paste it in the report, means it was thoroughly audited during the conduct of this experiment, and the facility is also as per that. So whenever an external regulatory agency comes for an audit, they can ask for this particular, and they look at the file, and if they see the certificate in place, means they know that proper audit, quality assurance audit was done by the concerned department. So it will help in their audit when they try to do, if this quality assurance certificate is there and it says as per the principles of GLP, means then it will be audited for the principles of GLP, means everything what we have discussed yesterday, those things they can ask for. So being a organogram, job descriptions, curriculum vitae, training records, calibration records, instrument logbooks, deep freezer logbooks, your freezer logbooks, your instrument HPLC or spectrophotometry or any other instrument logbook, your what do you call the master validation plan, master calibration plan, any of those things they can ask for if your report says that the study was done as per principles of GLP. If you don't put that certificate, then they don't even audit that report. So if you claim those study to be a GLP compliant, then only regulatory agency would be interested in doing audit of your facility purely from a GLP perspective, if it is for a non-clinical safety, non-clinical species. So that's the flow of GLP. As I mentioned, QAU audits, your quality assurance unit will audit all the critical phases, okay, of the study. Correct. And lastly, in just one or two slides, don't want to spend much time. This is just done yesterday, so it doesn't need any recap. But very quickly, preclinical testing. We have already seen an overview. Animal models in efficacy study, because we want to establish the efficacy of your compound. Assay development, animal PK, PD, everything in preclinical development, and dose escalation, proof of concept, large efficacy trials. All this thing goes on while clinical development is happening. Non-clinical safety efficacy, clinical efficacy testing, very important for assessing the appropriate schedule, also to justify the drug combination, right? But please remember the take-home message: in vivo, in vitro, non-clinical studies doesn't necessarily tell you accurate prediction of clinical efficacy. Of course, our efforts are always, we can try and be as accurate as possible, but doesn't necessarily prove a drug which is active in a non-clinical species will show same kind of efficacy when you go in human. PK we already talked about, 2 species, hours to days could be the duration, and we try and find out AUC, volume of distribution, Cmax, Tmax, half-life from the typical PK study. Safety we try to cover, generally it is single dose, and we try to capture cardiovascular, respiratory and CNS. Toxicology in detail we have discussed, from tissues, like when you dose them, finally at the end you will have the tissues or the blood samples, and you can do drug analysis, you can do biomarkers analysis, you can do histopathology. All those things can be studied as a toxicology. Doesn't matter you are doing a single dose, I mean, acute, subacute, subchronic, chronic study, whatever study you are doing, all these things can be easily covered within your toxicology study design. We also talked about the timing of the studies, like in preclinical study, what is the duration, what is the time exactly when it is to be done, and what clinical study is supported. So we talked about safety, TK studies, single dose toxicity studies. All these things are IND enabling studies, so it needs to be completed before phase one. And of course, it supports phase one clinical studies. Repeated dose again before phase one, because we want entire safety to be defined before we submit our IND application. So repeated dose toxicity study in a rodent and a non-rodent species should be in place in order to submit for IND application. If it's a pediatric application going forward, then a genotox or a reproductive tox, all those things should be definitely completed. Even carcinogenicity study should be completed if you're planning to go for a pediatric clinical trial. So these are the means and ways how we can ensure the safety of the drug before it can go for pediatric clinical trial. Because when neonates and in infants we are doing those trials, we don't want any risk. We should minimize the risk as much as possible. So that's why carcinogenicity, genotoxicity, all those study data should be in place before you go for a pediatric drug trial. So I'll just stop here, and this is the topic which we actually discussed in detail yesterday. That is good laboratory practice, but I think it must be completely refreshed in your mind. And tomorrow we'll take up new topics, and again on Monday we'll do a quick recap. I may not cover what I covered today. Around 50% of the classes I tried to cover on a very high speed. I'm sorry for that, but I'm sure the purpose is achieved, because here we have already covered all these slides in detail during last 10 days. The purpose was just to refresh that. So in Monday class, I will try to refresh and have a recap of the lectures which we did not cover today. Okay, so like laboratory animal in detail we have covered, about toxicology studies, about ADME studies, about in vitro studies, including solubility, octanol solubility, or plasma stability, or microsomal stability, or simulated gastric intestinal fluid stability studies. All those things we'll try to cover in Monday class, along with computer-aided drug designing, QSAR, and other any topics if they are remaining, especially the role of different disciplines. We will spend few minutes on discussing the role of different disciplines, including microbiology, pharmacology, pharmacy, chemistry, zoology, veterinary, biotechnology, all these people, and quality assurance people or clinical research people. So now all of you have understood what is their role, where they find themselves, where they can fit in the scheme of drug discovery development if they want to go for a research career. And we will just spend around 15 minutes discussing about that in our subsequent class. So with that, let me stop here. Thank you. Thanks a lot.
Thank you, sir. Today we have so many queries from students. The first query I had posted just now in the chat box. You can check it.
Yes. So, of course, we won't have practicals for a CADD. It may not be possible to do, but we may in future do a separate course where we'll do a practical class, but we will cover a few basics of computer-aided drug designing for sure within the scope of this course. Why do prefer analysis, pharmaceuticals, civics department in industries, and why don't prefer regulatory affairs department industries? Why do you prefer analysis, pharmaceutical? Okay, so see, the point here is, if you're asking this from an industry perspective, then of course in industry there are departments. There are separate departments for analytical development. I mean, pharmaceutics department, and there are regulatory affairs department are separate. Basically, if you're asking this question from an industry perspective, each industry, at least the major industries I know, they have separate regulatory affairs department, right? And they have separate analytical pharmaceutical departments. So this is not correct. Actually, they do have separate departments, and regulatory affairs. See, please understand, regulatory affairs doesn't deal alone with analytical. Of course, while doing analytical pharmaceutical development, formulation development, regulatory knowledge is required. But regulatory affairs includes everything. It includes patent, right? It includes regulatory agencies understanding, and almost every requirements of the regulatory body or the agencies can be covered within the regulatory affairs department. So that's why it is a separate department, and analytical and pharmaceutics departments are all separate, at least in industry, not from academia perspective. I think in academia there is no such scope to have a separate department. So that's why within the pharmaceutics department everything must be coming. But same is not the case with industry. Regulatory affairs department is important for industries while doing the job or not. It is definitely important. I hope I answered this question, because regulatory affair is one of the very important department which takes care of the patent infringement of all the products which you're working on. And generally I have seen they work a lot on regulatory guidelines also. I mean, they have access, they have knowledge of all those regulatory guidelines, and they give their inputs to scientists. Because as a scientist, I could be a biological scientist or a chemist or a biotechnologist or a microbiology, but regulatory affairs is important department who should be able to guide each of these different disciplines with the applicable regulatory requirements, so that as a scientist I can align myself to those applicable guidelines. Is there any scope for regulatory affairs department industries? Again, I think same question, huge scope. Definitely there is a scope, but you have to you have to go to an industry where they have got a separate regulatory affairs department. But from a scope point of view, as I'm again emphasizing, each and every industry, a good industry, big industry, have separate regulatory affairs department. So definitely there is a scope. In vitro studies, how the drug is metabolized. So in vitro studies, how the drug is, because as I mentioned, we are using hepatocytes, right? So in hepatocyte, what happens is there is CYP450 enzymes expression. So those enzymes will actually metabolize your drug. So not all in vitro studies. Many people say that they're using cell-based models, okay, like permeability, canine adenocarcinoma cell lines, or made in Derby canine kidney cell lines, or other cell lines, and they say that we are studying the metabolism. That is wrong, because those cell lines, the expression of cytochrome P450 enzymes is minimal to negligible, or many times it is not even expressed. So with that negligible expression of cytochrome P450, metabolism in those cell lines cannot happen. So if you are using those cell lines, and if you are considering that you are studying metabolism, it is wrong. So use hepatocytes where there is abundant expression of cytochrome P450 phase 1, phase 2 enzymes. So that's where this is what we have discussed. Even enterocytes. Why enterocytes in vitro study? Because there is cytochrome P450 isozyme expressions, 3A4 evidently expressed, and more than majority of the drugs are metabolized by 3A4. So it's a best model. So in vitro metabolism can be studied, but in a right model, which has to be hepatocytes or enterocytes. Okay. Any other question?
No, sir.
All right.
Students, anyone having any queries, please put in a chat box. Sir, one more question.
Please.
Is there any scope for Pharma D students in drug discovery? And
Yes, yes, definitely PharmD students, there is a scope. And let me just tell you, I mean, as I said, that we will spend around 10 15 minutes on any of these classes, remaining classes, purely talking about the scope. Okay. So we will cover that in that discussion. But apart from that, as I mentioned, and let me re emphasize, chemistry, pharmacy, microbiology, biotechnology. Within the pharmacy, when I say, it includes B Farm, M Farm, your PhDs, or PharmD. All of you guys have huge scope in drug discovery and development, because, as you may realize, see, this is a even a generic market is within the purview of drug discovery and development. Okay, what all BA/BE studies we do for generic molecules, even that is captured here, right? So that's why I say that there is a huge scope. As throughout this lecture series you have seen that your knowledge from your each of these fields comes into the play at some or the other phases of drug discovery and development. So PharmD has also a role to play, and they do have scope to get into drug discovery and development. All right. So ANDA, abbreviated new drug application, also requires GLP. Yes, answer would be yes, because you need to have. See, whenever you go for this submissions, please remember the non-clinical safety data should have been done in a GLP environment. So those studies should be as per the principles of GLP. Take it for writing. So even if you're going for ANDA submission, also at least the non-clinical safety data should have been generated as per the principles of GLP.
yeah, that's also no more questions.
All right. So thank you, and I hope it was useful. And let us connect tomorrow, and I'll try
Yeah.
to cover all the pending topics
Okay.
within tomorrow's class, so that we complete the course. And then Monday we can do a very quick recap of today. We have done one recap, and the next recap for all the remaining topics we'll do on Monday. Okay, and Tuesday 12th you will have the assessment.
Yeah.
So
Okay.
thank you. Thanks a lot.
Thank you, sir. Thank you a lot. Students, I have given the feedback form in YouTube and Facebook also. Please check it and give that. Based on that only, I will provide the certificates. Don't forget to give the feedback form. Please check the feedback form. Please check the feedback form. I have posted in Facebook and YouTube also. Please check it and submit the feedback form. Based on that only, I will provide the certificates. Okay. Thank you, everyone. Bye bye.